project was looking at antimalarial medication. The reason for this is that malaria continues to be a public health epidemic that affects over 200 million people per year, with 3.4 billion people considered at risk for the disease and 1.1 billion people considered at high risk, according to the World Health Organization. Malaria has a mortality rate of 30%, with 90% of these deaths occurring in Africa, as seen on the map on the right. The main treatment for malaria is taking antimalarial medications. However, there's a high prevalence of counterfeit and substandard antimalarials. Counterfeit medication is a medication that does not contain the active pharmaceutical ingredient or API that it should, and substandard medications are ones that do contain the correct API but have been stored at inadequate storage conditions or exposed to inadequate temperatures and are no longer considered effective. So I wanted to focus on this problem and determining the way for identifying counterfeit medications. There are several current devices that are able to distinguish between a counterfeit medication and a standard medication. One is TrueScan that uses Raman spectroscopy, which is a very expensive technique. Another is GPHF Minilab, which uses chemical reagents that are also expensive and require extensive user training. And the last one was actually created here at Boston University in the Zaman lab, which is PharmaCheck. It utilizes microfluidics to give quantitative results, but it uses chemical reagents that require user training and also end up destroying the drug that is being tested. So wanting to create a device that could be used specifically in low or middle income countries where there is a higher prevalence of antimalarial medications, my group looked at creating a device that could be low cost, portable, and user friendly. And in order to achieve these specifications, we thought that using a device that used optical spectroscopy methods could be able to distinguish between counterfeit and standard antimalarials. So our project names, our first one was that we decided to use elastic scattering spectroscopy, or ESS, and fluorescence spectroscopy. There are two methods for determining the optical properties of drugs and using those to distinguish between a counterfeit antimalarial and a standard malarial pill. For our project, we used one brand of antimalarial medication, camusinate, which consists of two different drug types, artesunate and dimodocin. And the benefits of these two spectroscopy methods are that they are non-destructive to the medication that's being tested. After doing several experiments to verify that these optical methods could be used for this application, we then focused on designing a device that would be low cost, portable, and user friendly. So some background information on the two optical spectroscopy methods we chose. The first, ESS, or elastic scattering spectroscopy, is when light is passed through a multimode fiber, which is placed directly in contact with the surface of the sample that's being analyzed. A secondary fiber is used to collect diffusely scattered photons from that surface, and they are then analyzed using a spectrometer. For our case, in order to measure the elastic scattering spectrum of a drug sample, we cut the pill being tested in half using a razor blade and placed the fiber on the inside surface of the sample. The second optical method we used was fluorescence spectroscopy. In this case, we were looking to see if the API of a sample might be fluorescent and what those properties could tell us about the amount of API in that sample. So we're looking to see if we can use fluorescence spectroscopy to distinguish between drugs with different APIs, or to see how the APIs differed compared to substandard and standard antimalarials. As I mentioned before, both these methods are non-destructive, require minimal training, and are relatively inexpensive. In order to verify whether or not we could use these two methods to detect counterfeit antimalarials, we had to create a counterfeit model and substandard model for the antimalarials. We did this for the counterfeits. We took 4 drugs that had different active pharmaceutical ingredients compared to the 2 antimalarial drugs, artesunate and dimodocin. And for the substandard model, we took 2 standard antimalarial drugs and exposed them to a water bath at 60 degrees Celsius for 48 hours, which is outside of the optimal temperature range that these drugs should be stored at. So this model is based on previous literature that had been done. So we are very confident that we were able to model what a substandard pill would be exposed to inoptimal temperatures. So we first took our counterfeit models and used elastic scattering spectroscopy, seen on the left, to compare the optical spectra of the counterfeit pills compared to the artesunate and imodocin. You can see on the graph that the spectra are visibly different between the 4 different counterfeit pills and the 2 antimalarial pills. Using a dimensionless parameter, which is calculated using the ratio of the maximum emission intensity to the excitation intensity for each spectrum, we were able to quantify the spectral difference between the counterfeit pills and the standard antimalarials. And it was able to determine that there was significant separation and distance between the 4 counterfeit spectra and the 2 standard antimalarial spectra. We then also tested this counterfeit model using fluorescence spectroscopy, as seen on the right. Once again, there is a visible difference in the spectra between the distinct maxima that each spectrum has and the differing slopes and amplitudes at various wavelengths. Using the same parameter calculated previously, we did statistical analysis and found that for artesunate, we were able to distinguish all of the counterfeit pills except for one were statistically different from the artesunate, standard, antimalarial, and similarly for imodocin, all but one of the counterfeit models were statistically significant in their difference compared to the imodocin spectrum. So using both of these methods, we were able to distinguish between the standard antimalarial drugs and the counterfeit models that we used. We then looked at our substandard models, again using both ESS and fluorescence spectroscopy to determine what optical properties and spectra were for those sample models. Seen on the left is the ESS data using similarly the same parameter and statistical analysis process that we used for the counterfeit medications. We were able to see through ESS that we were able to see statistical difference in the spectra of the stressed artesunate drugs versus the standard artesunate antimalarial. However, we did not see any statistical difference using ESS between the stressed imodocin sample and the standard imodocin sample. However, using fluorescence spectroscopy, we were able to see the opposite. So there was statistical significance in the spectral difference between stressed and standard imodocin samples, whereas with the fluorescence, we did not see any statistical difference between the stressed and standard artesunate drugs. So combining both of these methods, we were able to identify and distinguish between the standard and stressed artesunate and imodocin samples, which is one of the reasons why we decided to incorporate both methods in our device to provide a 2-layer differentiation system. So the layout of our device design, we ended up not being able to fully build this device, but this is what we were expecting it to look like. In the first image, you can see the red drawer. That's where the user would be able to pull out a drawer to insert the pill that was being tested and insert it back into the device. And then there's a lever so that the user can position the probe inside of the device, and a touchscreen to display the results. So that's all the user would be interfacing with. All of the measurements and calculations would be done inside of the device. As you can see in figures C and D, we have the main parts of the device are the battery, the spectrometer, laser, and white LED to actually take the 2 different optical spectroscopy measurements. And we also have 2 different microcontrollers that will relay the spectra to the touchscreen where it will be displayed for the user. This is what the graphical user interface would have looked like. So this is what would be displayed on the touchscreen and what the user would be interacting with. You can see on the left that they would be able to decide which optical spectroscopy method they wanted to use to measure the drug, either ESS or fluorescence spectroscopy. They could then save the spectra and perform the statistical analysis on the data collected, after which the resulting spectra would be displayed, and the user would also see whether or not the drug was counterfeit or not. So the user would get real-time information on whether or not the drug would be safe for a patient to take. So in conclusion, this project showed that these 2 optical methods could be used to determine the quality of a drug. We were able to plan out a device that would be low cost, portable, and user-friendly. The device would allow patients and doctors to receive real-time information on whether a drug is counterfeit or not, and therefore helping to eliminate the problem that counterfeit medications cause. I'd like to thank everyone in the Biomedical Optics Lab, especially Professor Urban Biggio, and thank you for listening to my presentation.